Hello everyone, our names are Vivian and Nadine, and today we will be presenting our discussion on the topic of DNA fingerprinting and its use and significance in forensic science.
DNA fingerprinting, which is also referred to as DNA genotyping or DNA profiling, serves a variety of purposes. This technique can be utilized to identify lost or missing family members, determine an individual's heritage, and in law to prove innocence or condemn suspected criminals. This last purpose is the basis for DNA fingerprinting in forensic science. Forensics is the application of science used in cases of crime and civil law. However, forensic science is mainly concerned with the criminal side. On a deeper level, the purpose of DNA fingerprinting in forensic science is to examine DNA strands or samples recovered from crime scenes and identify differences and patterns between different strands and against a prototype.
The application of DNA fingerprinting in forensics has allowed for the comparison of molecular genetics found in an individual's DNA to that of another individual or retained crime scene evidence of a biological nature. This technique essentially provides scientific proof of an individual's presence at the scene of investigation, thereby rendering conviction or exoneration. Armed with an investigative tool of this caliber, has enabled forensic investigators to successfully solve crimes which would have in some instances otherwise remained unsolved or in other cases incorrectly incarcerated the wrong individual. In terms of scientific development, the technique of DNA fingerprinting was considered a fairly new advancement in the branch of molecular genetics, with the introduction of its use in the field of forensic science. Its discovery in 1984 by Sir Alec Jeffreys occurred in one of those light bulb moments in which he recognised that DNA contained distinct characteristics which existed on an inherently individual and non-repeating genetic level, aside from those of identical twins who represented the one exception. This technique he then termed DNA fingerprinting. Jeffreys' discovery of DNA fingerprinting was made possible with the use of the polymerase chain reaction, or PCR, a process invented by Kary Mullis in the 1980s. The mode of PCR involves in vitro application, whereby small numbers of specific DNA segments are significantly amplified through enzymatic synthesis. This method, along with the earlier discovery and model established by Watson and Crick regarding the structure of DNA, allowed Jeffreys' technique to evolve. His revolutionary method would see the field of forensic science thrust into the public arena when it was to debut in its first ever forensic murder investigation, which led to the arrest and incarceration of the correct perpetrator and subsequent exoneration of an individual who was incorrectly convicted of the crime. This case would prove the fundamental use and utmost significance of DNA fingerprinting in the field of forensic science. However, as advancement in technology continues, the techniques employed for DNA profiling also advance, and had therefore seen Jeffreys' initial technique for DNA fingerprinting become obsolete and replaced by updated models such as PCR, short tandem repeats, and mitochondrial sequence determination. DNA fingerprinting has become a well-known concept in recent years, largely due to television programs which focus on crime scene investigation, as well as the news and media. But how exactly does it work? Approximately 99.9% of DNA can be shared between humans, leaving the remaining 0.1% of DNA which attributes to what makes an individual unique. Much like the rich patterns found in our fingerprints, our DNA is a genetic fingerprint which cannot be replicated unless in the case of identical twins. In order to successfully identify a person by their unique individual genetic code, DNA is required to undergo testing, namely PCR, to detect the incomparable banding patterns within that individual's genome. This requires a biological sample of DNA to which enzymes are employed to produce DNA of varying lengths. Once cut, these pieces are processed by gel electrophoresis, whereby the DNA is loaded onto agarose gel and an electrical current is passed through to sort the DNA according to size. These are then transferred to a nylon membrane for incubation, which involves labelled radioactive phosphorus probes that attach to complementary DNA within the genome. Exposing these to X-ray film allows for the unique banding patterns to be identified. By applying several DNA fingerprints to the same agarose gel, forensic scientists are able to compare biological evidence found at a crime scene to those of the suspects, resulting in the correct identification of the perpetrator. Since the development of DNA fingerprinting, its potential uses and significance across a vast and varied number of scientific fields has become apparent. DNA fingerprinting, which was initially used in samples involving human DNA, has seen current applications of the technique extend from forensics and identity determination through to medicine and medical diagnosis and paternity and immigration testing. With such broad applications of the technique, its potential use outside of human DNA was recognized and has enabled DNA fingerprinting to be employed in such diverse areas as botany, zoology, archaeology, and anthropology. This technique has, without question, proven to be an invaluable resource and tool for science. With the successful emergence of DNA fingerprinting into forensic science and identity determination, its importance in medicine would soon follow. Today it is used for the discovery and identification of diseases, as well as conditions inherited from predecessors. Its clinical importance is further highlighted in the assessment of donor hematopoietic engraftment after bone marrow transplantation and chimerism analysis after solid organ transplantation. It has further proven its worth in the resolution of paternity and immigration disputes, in particular the 1985 immigration case involving an Indian boy who would later have his case proven via the application of Jeffrey's DNA fingerprinting technique. Other fields of science which have greatly benefited from Jeffrey's technique include those of zoology and botany. Through DNA fingerprinting, zoology and the conservation of biodiversity have been provided with a means for observation and therefore the power of law enforcement to help secure conservation measures. Botany has also advanced through DNA fingerprinting, with its importance as a tool in genotype identification and plant ecology proving invaluable. The multitude of areas to which DNA fingerprinting can be applied to, and the scope of potential it has presented them with, has been fundamental in their evolution and their continued advancement.
Using the polymerase chain reaction in DNA fingerprinting holds its advantage over other methods as it is more accessible for general use. It is also a process that is relatively easy to undertake in laboratories, on top of being relatively quick compared to other methods such as the variable number tandem repeat, or VNTR for short. A VNTR can take weeks before the results can be determined, compared to 24 hours using the polymerase chain reaction. This comes in handy when court cases need evidence so that they don't have to wait for long periods of time before executing justice. With the polymerase chain reaction, only small samples, such as fragments of hair or skin, are needed to perform the process, which is useful at crime scenes when little evidence is left by criminals. Unlike other methods, at the end of the polymerase chain reaction, the samples can be reused to carry out the process to make sure the results can be duplicated and to eliminate the chance of error in the process. When the entire process is carried out correctly, the results are highly reproducible. The polymerase chain reaction can also accurately identify many alleles in the samples provided. Alleles are mutations from the original gene they deviate from. Other methods have their own advantages, such as the variable number tandem repeat, which can use large samples and is more accurate than the polymerase chain reaction in the long run. Even though polymerase chain-based reactions have quite a number of advantages, like any technique, they also have pitfalls. Polymerase chain reaction-based techniques cannot use large samples of matter for the use of the process, which means that the matter has to be broken down or it is unusable altogether. Any contamination of the sample due to bacteria, radiation, or other sources may lead to error in the amplification during the process, which will ultimately lead to the results being inaccurate and so misleading. If the process is carried out incorrectly, the results will be affected. Time also brings with it degradation of the sample, so any tests have to be carried out sooner rather than later to ensure accurate analysis. This means that storing the sample for later work, in the case of re-examining evidence, is near impossible. Unfortunately, polymerase chain reaction-based methods cannot identify all alleles in a sample, and their distribution is not flat, which means that more loci are required in the analysis of DNA samples. This method is also prone to error even if the procedure is undertaken correctly, which can lead to decisions that are erroneous and lead to the conviction of the wrong people. Other methods take longer to complete and are still prone to error. One of the major problems with every technique in DNA fingerprinting is that they cannot distinguish between relatives, such as siblings or cousins. In the future, DNA fingerprinting may one day be able to analyze samples of DNA collected and construct an image of what the person may look like without prior knowledge. This would remove countless searches and retrieval of DNA from multiple persons, and so speed up the process of identification and apprehension of the relevant people. Furthermore, it will also enable the error in identification of suspects to be reduced, clearing innocent people's names. The detection and accurate identification of mutations is also another extension of the existing technique. There will of course always be ongoing development of existing techniques to refine and remove limitations, such as what has been previously mentioned. The discovery and application of new techniques to the field of DNA fingerprinting will also be an ongoing part of forensics. This will mean that methods that are quicker, easier, and have less room for potential mistakes will be discovered and put into practice. As we have discussed today, DNA fingerprinting is an important and fairly recent tool in forensic science. It is used to deliver justice and lift suspicions from innocent people. It was developed quite quickly and put into use in the realms of justice, and has since risen as a key tool in forensics. DNA fingerprinting's roots are firmly planted in biochemical concepts at a molecular level. Its various methods have different strong and weak points, and like most other branches of science, it is constantly under review and further research is always being undertaken to discover new applications and methods and to eliminate error and create a more efficient process. It has come a long way and there is still more progress to be made. Thank you for listening to this presentation.